Good evening, everyone. Welcome to the second session of the SDSU Native Plant Initiative research updates. We're very excited to hear from two more graduate students who are working on research related to the Native Plant Initiative. My name is Christine Lang, and I'm an assistant professor and SDSU Extension Consumer Horticulture Specialist based in Brookings with statewide responsibilities. I want to give a special nod to my event co-planners, Drs. Krista Ehlert and Laura Perkins, both of whom are faculty in the SDSU Natural Resource Management Department. For a little bit of background on the NPI, the SDSU Native Plant Initiative was formed to highlight and elevate the award-winning and internationally recognized work done on native plants across the region. The mission of the NPI is to conduct research, education, and outreach to support excellence in native plant restoration and production in the Northern Great Plains. And we will be happy to share both a website and social media links so that you can read more about the Native Plant Initiative and stay tuned to all of the happenings behind the scenes. It is my great pleasure to welcome our first speaker for tonight's program. We're going to hear from both students for approximately 20 minutes. We're going to have time after each student presenter to have Q&A. So any questions you have this evening for either Gabby or Greg, please put those in the Q&A box at the bottom of your screen, and we will address as many questions as we can. And I'm sure both students will be happy to share contact information, and you'll also be able to contact them through the NPI social media site as well. So our first presenter this evening is Gabby Bulwark. She is a graduate research assistant in the Department of Natural Resource Management. She has her bachelor's degree in soil and land management from the University of Wisconsin-Stevens Point. Between her undergraduate and graduate studies, Gabby worked as a soil conservationist with the USDA Natural Resource Conservation Service in Haiti, South Dakota. Everyone, please give a virtual round of applause and welcome Gabby as she presents on the effect of agricultural chemicals on native plants in the Western Corn Belt. Gabby, the floor is yours.
All right, thank you, Christine. I will share my screen here. Okay, let me. Oh, come on. Hang on.
No worries. We'll give you a minute to settle in. No problem at all.
Okay, here we go. Okay, and you still see the same screen?
Yep, everything looks good on my end.
Perfect, thank you. Sorry about that. So yeah, as Christine said, my name is Gabby Bulwark, and I'll talk a little bit about my research on the effect of agricultural chemicals on native plants in the Dakotas. I'd like to start off by talking about the importance of grasslands and prairies. So they provide a lot of services for us, such as climate regulation, wildlife and pollinator habitat, food sources, water quality regulation, recreational opportunities, erosion prevention, and cultural services. Unfortunately, we are seeing a loss of grassland due to conversion to different land uses, mainly to agriculture. And the loss due to conversion then decreases the services that we can get from this land. And conversion in the Dakotas is estimated to be about a 72 to 99.9% of grassland lost. So that's compared to our historical grassland amounts. So here we've got a graphic of North Dakota's land use trends. So on the left-hand image, you have the corn and soybean trends. So the blue color is an increase in that land use, and the red color is a decrease in that land use. So as you can see, sort of on the right-hand side of that graphic, you've got some increase in the corn and soybean land use. So that means more of that land use is being farmed there. And then you can look at the grassland trends, which are on the right side, and that red color is the decrease of that land use. So we're seeing losses of grasslands kind of in some two big patches there. But one sort of hopeful thing is we do see some of that blue color in our grassland trends. So that means that we are gaining some back, but currently the loss is outpacing the gain of grasslands. So that leads to what we call fragmentation, which is when contiguous grassland is broken up and isolated from other grassland. That also makes it more likely that the grassland is adjacent to agriculture. So as you can see in the image to the right, we can see clear signs of agriculture, right? So those circles are from pivot irrigation. You see some of those, really defined rectangles. That's also very likely agriculture. And then you do see some of these green spaces, but they are not connected to each other. So that's what we call fragmentation, is when those grasslands are separated. Now, fragmentation can have some consequences. It can cause low quality habitat for wildlife and for plants. It can also increase the likelihood of non-target chemical exposure, which is when a chemical is affecting an organism that is not meant to be used on. It also causes limited movement for organisms. So that's your wind dispersed plants, or if you have organisms with a limited range, such as, we have some endangered butterflies like the Dakota skipper and the Poweshiek skipperling. It can cause a disruption of their movement corridors. That can also reduce the gene flow in those populations, so we get a reduced genetic diversity as well. Now, we can sort of combat this with ecological restoration, which is recovery of a damaged or degraded ecosystem. And this is commonly performed using seeds, so seed-based restoration. And that is when you go and plant the seeds of your desired species that you want to see in that area. And this can be used to convert some of that agricultural land back into grassland, and therefore decrease that fragmentation that we're seeing. Although it is estimated that about 19 to 49% of restorations fail, and a failed restoration is one that is dominated by exotic species instead of the species that you would like to see. So this leads scientists and managers asking, why is this happening? Why are we seeing these failed restorations? So what we're trying to test out is to see if pesticide drift might be affecting our restorations. So pesticide drift is movement of pesticide droplets or dust through air at the time of application or soon after to any site other than the area intended. So liquid application of pesticide is known to drift. It can drift up to 1% out to 20 meters away from that target area. And it may impact non-target seeds and seedlings, even at those small doses that you see with drift. So you can see in the image to the right, we have a tractor with a boom sprayer there. Those are the kind of wings sticking out of the side that is spraying a chemical onto the field. And if you can imagine, if you have a particularly windy day, those droplets can get blown away and, onto a different field. So we are going to study 3 different herbicides, which is kind of a subset of pesticides. So this graphic comes from the US EPA. It is the most current graphic they have on herbicide use on their website, which is why we chose this one, even though it's from 2001. And we decided to use some chemicals in the top 5 of most frequently used in the US. So we are going to use trifluralin, 2,4-D, and atrazine. We decided not to use glyphosate or dicamba because of some conflicts of interest. So now I'll talk a little bit about each of the chemicals. So there's sort of 2 ways that these herbicides can work. So there's pre-emergence and post-emergence. So a pre-emergent is an herbicide that acts on like a seed or seedling before it emerges from the soil, so before it breaks the soil surface. And a post-emergent is one that works on a seedling once it has come out of the ground. So atrazine is both a pre and post-emergent, and it controls both broadleaves and grasses. It can be applied at a wide variety of times, and it is surface applied, so like by the boom that I showed you in the previous picture. or it may also be incorporated into the soil. And then we have 2,4-D. It is a post-emergent, and it controls emerged broadleafs. And it can be applied before planting your crop or after the crop has emerged, and it is also surface applied. And the example of damage that we see here on the right is 2,4-D damage on a grape leaf. And then trifluralin is a pre-emergent, and it controls both broadleaves and grasses. It can be applied at a wide variety of times, and it is also surface applied or may be incorporated into the soil. And in the photo, we see trifluralin damage on a tomato plant. So here are the species that we chose to study. I have 15 different species listed here, and we selected species that have a range of success in restorations so that we can hopefully also see a range in their reaction to the chemical. We also wanted to choose both grasses and forbs since both are used in a restoration setting. And we chose species from different families as well. So our first objective for this research was to evaluate the effects of agricultural chemical presence at different concentrations on native seed germination. So our hypothesis, what we think is going to happen is that as chemical concentration increases, we'll see a decrease in the germination rate. And germination rate is how many seeds per day sprouted, and final germination percent. Final germination percent is the total number of seeds that germinated by the end of the study out of the total that we started with. Our second objective is to evaluate the effects of agricultural chemical presence at different concentrations on native seedling emergence and growth. And our hypothesis is that as the chemical concentration increases, we would see a decrease in the seedling emergence, which, as I stated before, is when the little seedling comes out of the ground. We would think we would see a decrease as well in the leaf count, the height, and the dry biomass, and that we would see an increase in seedling death once they have emerged. Now I'll share my methods. So this is how I conducted the experiment. So for our germination study, first I had to make our different chemical concentrations. So I started off making the 100% solution, which is equivalent to what a farmer would be spraying on their fields. So that is our 100%. And then from there, I diluted it down. So then we have a 50%, a 10, a 1, and a 0.1%. And we used just water as our control. So that range in the middle there is the range of effects that, or the range of chemical that we might see at varying levels with pesticide drift. Then our step 2 is we would dip some germination papers in chemical. Germination paper is just essentially fancy brown paper that we use as a medium to put our seed on, and it holds water. So we would dip those in our chemical and then place that paper into a resealable plastic bag, once again to keep the water in with those seeds. Then we would put 25 seeds into each bag and then count the seeds for germination. So as you can see here, we have circled the germinated seeds, and we consider a germinated seed one that has a radical or looks like a little root sticking out of it. And then the different colors of Sharpie that you see on those bags just represent different days that those emerged. We just use colors to keep track of the days in case we got something a little bit mixed up. And now for the emergence and growth methods. Our first step was to prepare our tubes with soil and then plant seeds in each of those tubes and then cover them with a little bit more soil. And then I had to mix the chemical and apply it to the tubes. So I mixed the chemical like I did in the other experiment. we had our 100%, and then with the emergence, we skipped the 50 and we did the 10, 1, and 0.1 with 0 as our control. And I applied them to the tubes with a pipette, which is that instrument that you see there. And it just dispenses the same amount of liquid into each tube. Then we would track emergence of the seedlings every day. And then once they have been emerged for 7 days, I would measure their height and count how many leaves they had. And then once they've been emerged for 28 days, I would clip them and dry them and take their biomass so that we can compare that between our treatments. So in the photos here, I've got examples of what different seedlings look like once they've emerged. So we do not have data for our greenhouse emergence studies yet because those are still ongoing, but I do have data for our germination studies. So that is what I'll share with you today. So we do see trends with the atrazine and trifluralin exposure, which were 2 of our chemicals, but it was not as strong or widespread as what we saw with the 2,4-D. So I'm going to share the 2,4-D results with you because they're pretty interesting. So here we're going to look at the mean germination rate results. So once again, mean germination rate is the average of how many seeds germinated per day. So we found that 13 out of our 15 study species were affected, that we saw a decrease in our mean germination rate. So on the graphs on the left, you can see they have sort of this stair-step pattern. So along the bottom, they're labeled the 0 through 100. So those are our chemical concentrations. So as you can see, the lighter colored bar, which is our 0%, has the highest mean germination rate for each of those species. And then you see the stair-step pattern, or you see the slope going downwards, which means that we see fewer seeds per day germinate, the higher that chemical concentration gets. So that means that your seeds are not going to be coming up, all at the same time, or even coming up necessarily at all, if they're exposed to some of those higher levels. So these 4 are just an example of the 13 species that were affected. I didn't feel the need to share all of those with you because it's a lot of information. So these species that were affected, some of them were purple prairie clover, wild bergamot, gray-headed coneflower, and Indian grass. Now the species that were unaffected were western wheatgrass and green needlegrass. Then we'll look at the final germination percent results. You can read the graphs the same way that you did on the last slide. We have our concentration along the bottom and then our final germination percent along the side. So once again, we have 13 out of 15 species affected, but they were a little bit different. It wasn't the same species affected or unaffected. So once again, we can see that stair-step pattern or that sharp decrease in final germination percent. So this means that, if we look at smooth blue aster, which is in the bottom left corner, we see about 70% germination in our control. So that means if you plant 100 seeds, about 70 of them would come up. But if they were exposed to even 1% of 2,4-D, only about 20 out of those 100 seeds would germinate. And that's a really big difference to be affected by 1% of a chemical. So smooth blue aster is especially sensitive to 2,4-D exposure, even at small doses. Same thing with the Indian grass, which you can see in the graph above it. So the species that were unaffected were switchgrass and purple prairie clover. So what this all means is that we would recommend for tolerant species to the chemical that you could plant western wheatgrass, green needlegrass, switchgrass, and purple prairie clover. and that should be a pretty safe option for you. We only tested this at the germination and emergence stage, though. There may be effects to adult plants that we have not tested, but for those life stages, these species should be pretty safe for that exposure. And we also recommend that anybody that is spraying chemical follows the laws and regulations surrounding it. It's also really important to pay attention to weather conditions. If it's too hot or if it's too windy, sometimes that can cause it more likely that you'll have drift when you're spraying. There are also drift control agents, things that you can do to your equipment or things that you can add to your pesticide mix that makes it less likely for drift to occur. So with that, I would like to say thank you very much, and I can take any questions.
Wonderful. Thank you, Gabby. And if you want to leave up your contact information for just a minute, I think that'll be really helpful. You wrapped up with that extended comment on drift. I just want to share that one of tonight's listeners shared that drift can be more than droplets. It can also be volatilization, ambient in the atmosphere, and via soil erosion, which is a really good point. And I feel like you touched on that. Do you have anything else you'd like to say on that?
Yeah, drift can also, it can move through water. There's a lot of ways that it can happen, but what we were trying to target was mostly that droplet exposure.
And Gabby, maybe for our listeners, would you go back one slide to that summary of some of the ones that were most, or the tolerant species?
Yes.
And someone had a clarifying question about what were the species that were most affected based on the results you have thus far?
Most affected. Okay, so I would say the smooth blue aster and the Indian grass were probably the most extreme, at least for the exposure to 2,4-D. Otherwise, you see as we call it, the stair steps, right? So unless you're spraying directly on something you just seeded, you're not likely to see, the poor germination that you would get from 100% exposure. But it is important to know that if you're going to be planting a restoration that's near an agricultural field, you might see decreased germination levels from what you would be expecting.
Thank you. That's a really helpful take-home message. And again, we want to remind everyone that Gabby's going to be, you're continuing to do the trials, and you'll have more results coming out. So that's a reminder to pay attention to maybe the NPI Facebook page as results are coming out, and other places, or we'll have to have a version 2.0. So a few more detailed questions for you, Gabby. How long did you allow for germination before considering something to be non-viable? And their comment was, some of the carrot species I grow will germinate anywhere from 7 to 90 days.
So we tested out, so we had day 1 to day 45. If there was no more germination that was occurring on day 45, then we'd consider it done. But if germination was still happening, then we carried it out to 90 days. So some of these species we observed for about 3 months.
Excellent. Thank you. That's some dedication.
Oh yeah. Looking at those bags every day.
So the next question is, do those more tolerant species, do their seeds tend to have tougher seed coats?
Oh, that is an interesting question. I have not looked into sort of the mechanism why as of yet. I've just read my data. So that would be really interesting to look into. And I think, that's very valid. So I should look into that.
Awesome. Thank you for that question. And this is why we love doing research updates, because we walk away with new ideas for our research. All right, at this time, I'm not seeing any more questions for Gabby. So Gabby's going to stay with us all evening. So if anyone else thinks of another question, Gabby, we will bring you back at the end for open forum with Greg as we close out the night. But thank you so much.
Sounds good.
And
thank you.
We will let Greg get queued up. And I just want to remind people, keep those questions coming. And before you duck out of tonight's session, we certainly would love to hear your feedback and have you answer just a few questions about what you learned in tonight's session. So please watch for that in the chat. And it's my pleasure to introduce our second speaker tonight, Greg Cooper, who is originally from Connecticut, moved to Brookings in January of 2022. He went to the University of Connecticut, where he majored in ecology and evolutionary biology. And before coming to SDSU, Greg was working as a field researcher for the Connecticut Agricultural Experiment Station, where he worked on a mosquito trapping program, monitoring sites for West Nile virus. So everyone join me in a virtual round of applause as we welcome Greg to tell us more about determining germination requirements for native plants of the Northern Great Plains. Greg, I'll let you take it away.
Thank you for that introduction. So as Christine said, my name is Greg Cooper, and I will be talking tonight about my research on dormancy and germination requirements. But first, I want to talk about why I'm focusing on these native species, and it's because their habitat is in trouble. The World Wildlife Fund put together this plow print map in one of their reports, which outlines the entirety of the Great Plains. The green areas are what they estimate to be intact grasslands, while the dark orange is land that has been converted to agriculture. And this land conversion is happening at an alarming rate. Forbs that I am researching are coming from the Northern Great Plains, which consists of the Dakotas, parts of Nebraska, Wyoming, Montana, and up into Canada. And within this study area, over 500,000 acres of grassland were converted into farm each year from 2015 to 2019. Some studies even estimate that up to 50% of grasslands in the Northern Great Plains could already be lost. And there are huge losses that are coupled with the degradation of grasslands. We see declines in biodiversity, a loss of carbon storage. We see increased erosion, and we also lose a cultural identity. One possible solution into reestablishing these grasslands that we love is through restoration. We can take degraded lands, such as abandoned ag fields or landfills, and restore them into native prairies, which will increase local diversity, reduce fragmentation, and can be used for recreational purposes. These photos here are of the same piece of land in western Wisconsin. The photo on the left shows how the field was originally planted with soybean. Eventually, the owners decided to restore the land to grassland, and after only 4 years, we get the image on the right. However, not all planting and restoration projects are successful. A 2016 study determined that many restoration projects saw germination success rates even lower than 20%, which means 80% of the seeds they planted did not grow. This is likely due to our collective lack of knowledge on germination among species. Many large projects also use seed mixes and mechanically broadcast these seeds. This process gives no consideration into germination requirements of individual species. What we really need to identify is the best germination conditions that encourage germination for high priority species within these seed mixes. One mechanism that could be preventing germination is dormancy. Seed dormancy is a biological mechanism within the seed that prevents germination even in suitable environmental conditions. This means that even when there's plenty of water and light, there are still some underlying requirements that haven't been met yet, preventing the seed from growing. Although it can be frustrating for a gardener or restoration practitioner, dormancy ensures long-term survival of the plants. For example, by bet hedging or allowing seeds to germinate in different years, there is more chance of survival instead of having your seeds germinate all at one time to find out there's a drought and none will survive. What this graphic shows here is that a particular seed that has dormancy could have plenty of sunlight and water, or the suitable conditions I said earlier, and still not be able to germinate. Our plan is to use laboratory treatments to break these underlying requirements and speed up the germination process. And again, for restoration practitioners who need their plants to grow quickly, this can be an issue, and it can be a fairly complex issue. As it's estimated that about 70% of plants have some sort of seed dormancy, and that dormancy can be expressed in one of 5 different ways. I don't want to go too in depth into the different dormancy classes, but the real important takeaway is that they each require unique methods in order to break dormancy. For example, physical dormancy is caused by a barrier, such as a hard seed coat. That seed needs to be broken before germination. Physiological dormancy needs chemical or specific temperature cues. Morphological dormancy needs extended periods of time for longer embryonic development before it's ready. And if that's not enough, combinational and morphophysiological both require 2 dormancy breaking steps in specific order. But again, the takeaway here is that dormancy is fairly common in our native forbs, and it's not always easy to determine which methods are needed. So for my project, we have 2 research objectives. The first is to determine the best germination conditions. This will help the success of these species and plantings and restoration projects. We will look at if the seeds need specific temperature conditions or if they need to be treated before planting. Our second goal is to determine if the seeds express dormancy, and this will help to gain a better understanding of the germination mechanisms within the seed. So our plan was to put 15 species through a germination experiment to determine these requirements. Initially, the Bureau of Land Management provided me with a list of high priority species for restoration. And from this list, we decided to focus on forbs, and we selected 15 perennial species that are all native to the northern Great Plains. But before I talk about how I treated the seeds, I wanted to show you what my experiment looked like. From the picture on the left, you can see I used brown germination paper, which I would dip in water. I would evenly place 50 seeds onto each germination paper and slide that paper into a plastic bag before sealing it. Bags would later go into a chamber. And I checked the seeds about every 3rd day in the experiment. I would look to see if the seeds germinated, and like Gabby, if they did, I would circle them on the bag and record my data. However, before seeds were put into the bags, the first thing I did was give the seeds a pretreatment. Each pretreatment will give us a better understanding into if dormancy is present. For example, scarification should break the seed coat or barrier in any seeds with physical dormancy. And fertilizer would give chemical germination cues to the seeds and so on. So the purpose of the pretreatments is to simulate different environmental treatments that would promote germination in nature. Of the 4 pretreatments, the first was scarification. The picture on the top left shows the electric scarifier I used. I lined the barrel with sandpaper, and there are paddles inside the barrel that rotate. So the seeds get knocked around until they are scratched or the seed coat is broken. There are many ways to scarify seeds, such as nicking them with a razor blade, but this was the easiest way for the amount of seeds I was using. The second photo shows the fertilizer pretreatment. In this photo, you can see the sheets of germination paper I used. For all other treatments, the paper was moistened with water, but for this pretreatment, we soaked the papers in potassium nitrate before putting seeds on them. Due to the historical fire regimes in the Great Plains, we also decided to expose the seeds to smoke, as that has been shown to cue germination in some other species. So in our third picture, you can see we place seeds into a large cooler. I burned 1.5 grams of wood and pumped the smoke into the sealed container for 20 minutes. And finally, there was the control, in which the seeds were left untreated. After the seeds were given a pretreatment and put into bags, they were then placed into a germination chamber. The photo all the way to the left shows one of our chambers that we have in our lab. So this experiment was based on a concept called the move along experiment, where seeds rotate through different chambers of different temperatures to simulate movement through the seasons. And we had 3 chambers in our lab, one to represent summer, one for winter, and one for spring and fall temperatures. Each chamber alternated between 12 hours of light and 12 hours of dark. They also had daily high and low temperatures, which is why you see a range of temperatures on the slide. So for example, the summer chamber had a high of 29 degrees Celsius and a low of 16 degrees every day. And the temperature ranges were gathered from weather data around the northern Great Plains over the past 20 years. So in order to test for the best temperature conditions, I had treatments start in different chambers. Some would start in the summer and some would start in the winter chamber. I also staggered the starts of the treatments at 2, 4, and 8 weeks to simulate different planting times. Between all the different temperature regimes in the move along and the 4 pretreatments, we should gather enough information to allow us to determine the best germination conditions for each species. And it did give us results. So I won't share all of the species with you, but I want to give you a range of examples about what my research found. Starting with blanket flower, we found that the temperature conditions did not have any real strong effect on germination. However, pretreatments did influence results. This is a graph of the germination results for blanket flower by pretreatment. On the bottom x-axis, you can see bars for each of the 4 pretreatments. And on the y-axis is final germination percentage from 0 to 100. For most pretreatments, final germination ended between 60 and 70%. However, when I scarified the seeds, which is the orange bar you see, they germinated to almost 80%. And although there was adequate germination without treating the seeds, scarification increased germination by about 15%. So in this case, we would recommend scarifying blanket flower seeds. And as I said earlier, I also wanted to determine the dormancy of the species. What these results show me is that although a lot of the seeds are non-dormant, meaning they don't need any treatment, about 15% of the seeds are physically dormant, meaning the seed coat needs to be broken or scratched first. And this 15% number I give is by no means a baseline for all populations of blanket flower. All populations and seed batches will be a little different, and there'll be variation depending on if they're wild collected, commercially purchased, or where they originally came from. Our seeds were purchased commercially, as that is what large-scale restoration practitioners would be buying. And this information suggests to us that for purchased blanket flower seeds, you'll likely see better seeding success if you scarify them first. For a different example, I will show you the dotted blazing star results. For some species like this one, it was very easy to tell that the seeds were non-dormant. Again, temperature conditions did not have a strong effect on germination, and here neither did pretreatment. So I have another graph of final germination percentage by pretreatments. And as you can tell, germination was high for all of them. In situations like this, we would consider the seeds non-dormant, and our recommendation would be that the seeds are ready to plant as is. No special treatment is needed. And while data for species like dotted blazing star were very simple, Some species were more difficult to interpret. So I wanted to show you the results for white penstemon. With this species, pretreatment had no effect on germination. However, temperature did influence results. Here is a bar graph for germination percentage by treatment condition. On the x-axis, there are 9 different temperature regimes. The first 4 treatments, the treatments that start with C, all began their cycle in the cold or winter chamber. The number following is the length of stratification. So C2 was stratified in the cold chamber for 2 weeks. We also had 4-week, 8-week, and a treatment that was continuously in the cold chamber. This middle treatment, the one labeled NCON, had no stratification, and it stayed in the spring chamber for the entire experiment. And the last 4 treatments all started in the warm or summer chamber. Again, I want to highlight the statistically highest treatments in orange. And what we see is that some treatments that started in the winter and some that started in the summer both had high germination. So how do we interpret that? Do I want to plant the seeds in the summer or winter? Which is best? Well, what we need to do from here is look at when the seeds germinated. So what I did was graph some treatments over time. The experiment starts at the bottom left corner of the graph at day 0. And as time goes on, I graph a cumulative germination. Before, I showed you that all these treatments had high germination. And as you can see by the end of the experiment, in the top right corner, all treatments are at about 80% germination or higher. But the clear difference is the timing of germination. The blue lines, which are treatments that started in the winter chamber, germinated well before the red lines, which were the treatments that started in the summer chamber. So it's still kind of confusing. What is happening between the warm and cold treatments? Well, what I'm going to do is add a blue highlight to the graph. Each highlight represents when one of the summer cycle treatments spent time in the winter chamber. So if we look at the red lines, we can see that germination only begins to spike after spending some time in the winter chamber. We also see that the blue lines, which started in the winter, germinated immediately. Putting this together, we can tell that white penstemon needs a period of cold stratification in order to germinate. So if we go back to the bar chart, I would recommend, in this case, ignoring the warm treatments that did well, because what this chart doesn't show is that these warm treatments took a lot longer to germinate. Instead, I would focus on the cold treatments and recommend that white penstemon should be cold stratified for at least 4 weeks to get best germination results. And finally, I will show you the results of wax leaf penstemon. These results were extremely similar to those of the white penstemon. Again, there are some cold and warm treatments that both did well, highlighted again in orange. I'm not going to show you another line graph, but again, treatments in the cold cycle germinated immediately, while those that started in the warm cycle were severely delayed. The difference between the 2 penstemon species is that while 4 weeks of cold stratification was enough for white penstemon, clearly here the wax leaf penstemon needed at least 8 weeks to improve germination results. So at the moment, I'm only able to share the results of a few species, but our end goal is to analyze the results for all 15 species and bring this information to the public to help improve the success of seed-based restoration projects and gardeners working with these species. And I couldn't have done any of the research without help along the way. So I'd like to acknowledge all those who assisted with this project, especially my advisors, Dr. Perkins and Dr. Leffler. And I'd like to thank you all for joining today, and I'm excited to answer any questions.
Wonderful. Thank you so much, Greg. And if you'll stay on that page for a moment, and we've got your contact information. Thank you. I want to start by passing along a word of, wow, that restoration photo was wonderful. So we had an audience member who loved your photo. And that's a good segue into the first question. And if people have additional questions for Greg, please put those in the Q&A. And if you have thought of any additional questions for Gabby, please drop those in the Q&A as well. So Greg, the question was about your germination paper. Was that a special product or is it just brown paper? The person asking the question has used paper towels in the past with some success, but they had issues with mold. So what are your thoughts on germination paper?
So yeah, it's a special paper. It's, more textured than normal paper. As Gabby said, it holds water. It is quite similar to paper towels. I'd say it's a little bit thicker and it holds water a little bit better. But I'd also say that we also have issues with mold in this project. So I don't think that changing the paper would necessarily fix that problem.
All right, thank you.
And I can
ahead,
share, the brand we use is called Anchor germination paper. And it is a special paper that is used for seed germination, like across the industry. But I'd be curious to see if brown paper would do something different than paper towel.
So that would be an additional research study, right? So Greg and Gabby, well, if folks are thinking of other questions, I'm going to put you on the spot. What's been one of the most surprising things you found in your research? Like, what was something obscure that happened that was unexpected?
That is a good question. I think as far as the results, seeing a wide range was very exciting. Between results that I needed to dig deeper into the literature to understand, and others like the scarification that were straightforward. So it's fun to see a bunch of different classes of dormancy expressed, which I felt was exciting and unexpected with only 15 species.
Excellent. Thank you for sharing. Gabby?
I would say with my greenhouse experiment right now, I'm seeing some plants grow into some really weird shapes and different, the leaves are not looking quite like leaves should. So I'm curious to see what happens once they grow up a little bit more, and if they can kind of self-correct or if that's kind of the end for them. So I think, that's really interesting.
We'll look forward to seeing photos.
Yes.
All right, this has been great because a few more questions are coming in. So Greg, would you be able to share any insights on the temperature in the box when the seeds were exposed to smoke?
Unfortunately not. So we standardized that treatment by the type of wood, the amount of wood that was burned, and the length of time. So we didn't have the equipment in the lab at the moment to figure out the temperature in the box, unfortunately.
That's all right. Thanks for being honest and sharing how you standardized. So the next question for you, Greg, is would cold treating a bulk seed mix, if that's how they are typically sold, be a good catch-all strategy for increased germination, or could that harm other seeds in the mix?
That is a good question. I don't know if I have enough information on that to answer it fully. I would say so far that none of the species we've found have had negative results towards being in the cold storage first. Well, I shouldn't say that completely. I think some of them had a small decrease in germination, but ours was cold stratification. So it was in the cold chamber in a moist environment. And we're not completely studying cold storage, which can have a little bit different effects on it.
Wonderful. And Greg, the extension diplomatic answer is it depends. And you had a great, that was perfect. So another comment, and if you want to expand on this at all, please do. But it's interesting and a positive regarding the cold stratification, especially for those of us who garden that do winter sowing with many flowers. So winter sowing in the sense that maybe they're going out and putting that seed out on the snowpack or putting it out after snowfall or before snowfall and letting snow fall onto it. Do you have any comments on that for our gardeners?
Yeah, and I mean, I think that's, I'm glad I can present these results because that's what this information will be used for. And I think it's interesting seeing that species like waxleaf might need a longer amount of time, if you if you plant them out in the winter. And so I'm just glad that I can present those results to you.
Wonderful. Well, if anyone has lingering questions. Oh, okay. So some clarification. People wanted to clarify that winter sowing, and this is a trend that I am starting to see, is actually planting seeds into milk jugs and let the cold snow. So they're planting seeds in milk jugs in cold, snowy areas, and they're letting that snow get into the milk jugs. And you have kind of a mini greenhouse setting that affects plant germination. Any thoughts on how that might affect native plant germination?
Yeah, I'm not quite sure. I haven't actually heard of the technique. So that's something that I will do more research on probably tonight.
All right, excellent. I just learned about it a few weeks ago myself, but I know some of the gardeners in the region are trying to take advantage of doing that. So Greg or Gabby, based on conversations with stakeholders, what are some ways that you see the outcomes of your research being used? Or is there going to be like a handy dandy flip chart or a document of these are some great protocols? Could you expand on that for either of your projects?
Yeah, I'm hoping at the end of my project to be able to say, these are sensitive plants, these are more tolerant plants, and just be able to provide a recommendation list to folks looking to plant these seeds in areas that might be subject to some drift.
Yeah, and I'm working with the Bureau of Land Management. So all the species I had were plants and flowers that they used in restoration and had trouble establishing or germinating. So I'll give my information back to them with recommendations on how to better their success, and then publish my data to the public. So, private landowners can also use it.
Wonderful. So we'll see some impacts both on private and public lands, which is really, really exciting. So Gabby, we have a follow-up question for you. What evidence do you have about the impacts of herbicide drift on established plantings of native species? Or did you find anything in your literature review or through your project work?
Unestablished. So there's really not a lot of research about how herbicides are affecting our native plants. That's kind of a newer thing, which is why we are doing this. But I can't think of anything off the top of my head that shows unestablished species just with germination or effects on non-traditional crops or vegetable crops. Like there's a study on how it affects tomatoes, but that's not necessarily a native plant. So yeah.
Well, and I'm glad that you picked on tomatoes because tomatoes are very sensitive to so many herbicides that they're a great model plant is probably the wrong word, but they are a great plant to show all sorts of fun herbicide damage effects. So that's something I get to look forward to in my work. Greg, we have an audience member who is curious if there's a way for them to be alerted but when your final reports or results or recommendations go out, or will there be any sort of an alert on the NPI Facebook page, or could folks contact you and request that the public findings? Oh,
You're
and you
muted,
are on
Greg.
mute.
Thank you. Yep, I put my contact information on the last page, so if anybody wants to reach out to me, feel free to do so. And we will also make some sort of announcement on the NPI social media when those results are out.
Yes, I run the social media, hello. So if anybody in our lab, gets a publication, we'll post that so people can know where to find it.
And I should share, and we dropped the link to the NPI Facebook page in the chat. So if you haven't grabbed that, please do. Otherwise, just look up SDSU Native Plant Initiative on Facebook. But if I was to give an award for social media profile with the most beautiful photos, I think I would give it to you all. Absolutely gorgeous. And a great note, if folks didn't get Greg or Gabby's emails jotted down, that you can message NPI and you'll be interacting directly with the students. So as we're wrapping up, again, thank you all for your questions and all of your expression of gratitude and thanks. I really appreciate that. Gabby and Greg, if folks were interested in purchasing some native plants, would you mind giving a little advertisement for where they might look for you and whenabouts they could find you?
Greg, do you want to, or do you want me to?
Go ahead.
Okay, yeah, so we grow South Dakota native plants as a part of the Native Plant Initiative. So we're starting to plant those now in our greenhouse so that they will be a little more grown up by the time we sell them. But we are taking part in McCrory Gardens Garden Discovery Festival this spring, so you can find us there. We will also be out in Spearfish in June. I can't remember the exact date. And then McCrory also has like a fall festival that we will sell plants at. And we may be doing online orders again this year. So stay tuned to our Facebook page for that option if we decide to do that again. So the answer for questions is always going to be check our Facebook page because we'll post every kind of big event there.
Awesome. And thanks to you for that preview. The McCrory Gardens Discovery Festival is going to be Sunday, May 21st. So that's one that I can say with confidence that you'll be able to purchase native plants. And there will also be a Master Gardener plant sale at the event, in addition to all sorts of other great things. So also watch the SDSU Extension Facebook page for fun happenings. Well, as we wrap up tonight, again, thank you to all of you for completing the session evaluation. Would love to hear what you thought about tonight's presentation and what other native plant topics you might want to hear in the future. Perhaps we're going to have to welcome the students back later in the fall so we can hear more about your research findings and all of the things you've learned through further data analysis and looking at all of those results. So with that, thank you to all of our attendees. Extreme gratitude to Greg and Gabby for joining us tonight and so thoughtfully sharing their research work as well as giving us a preview of their results. And again, stay tuned to the SDSU Native Plant Initiative Facebook page so that you can find out about plant sales, releases of results, and just see lots and lots of gorgeous plant photos. So thank you all so much for joining us tonight and have a wonderful evening.
All right, thanks, Christine, and thank you, everyone.
Yeah.